All right, now let's dig a little bit more into what the content of the lab manual. After the first part of the lecture or lab lecture, now you should have a solid understanding about the basic principles of statistics, right? Now, um, I cannot emphasize more about like understanding what sampling error is, right? So in this particular figure, you are looking at different shell sizes and the number of times those shell sizes were found among a population of Asian shore crabs collected in last year in 2019, right? So here you're looking at the mean shell size of this particular sample was about 14.41 millimeters, right? And also most of the other samples collected here, right, have shell values, somewhat closer to it. But also notice that certain shells are extremely larger than the mean value, and certain shells are actually extremely smaller than the mean shell value, right? So the presence of these such extreme values, greater deviations from the mean can create problems with sampling error, right? So those extreme values does not help you to characterize the sample, and because of that, those extreme values are not representative of the entire population that you are interested in. Again, another way how you could commit sampling error, right? And now let's look at these two populations. Here I'm trying to again explain you the differences or the importance of properly understanding those sample statistics, right? So two hypothetical distributions of shell widths of Asian shore crabs are shown here to the left and to the right, right? Note that in the distribution on the left, right? Actually, let me get my pointer. Yeah, on the distribution to the left, right, the shell widths are much more tightly clustered than in the sample we have to the graph on the right, right? See the variation, like look at the mean, 15, 15. So both samples have the same mean, right? But in this figure, right, you can see the variation is, actually, around the mean is subtle, right? The minimum of 11 millimeters and the maximum of 19, with the mean being 15. Here the mean is the same, but the minimum can be as low as 3 millimeters. The maximum can be as greater as 27, right? So here the mean shell, the shell widths are tightly clustered around the mean. Over there, the shell widths are not tightly clustered, right? They spread widely around the mean. Okay, so because the sample depicted on the right has much more variable measurements, right, we call that particular sample to have a larger, greater standard deviation. This greater variability, right, can also result in a larger confident interval, right? It will have a larger upper and lower confident limits. So you're looking at, in that case, you're looking at a highly variable population, right? So it is much harder, much practically difficult to obtain a precise mean estimate, reliable mean estimate in a population that have such a greater variation, right? So if I call the left sample versus right sample, right? Left sample, right? I can, my sampling statistics, right? My sampling mean, my sampling standard deviation, sampling variance, those are quite helpful measurements, right? But to the right, right, because of this much wider variation, right, it's extremely hard for me to characterize that particular sample or that particular population, right, based on mean and standard deviation and variance, right? That is the point. Now it's really important, like particularly because of the reason I just highlighted, particularly when you actually cannot reliably depend on mean or averages, right? You need to compare the actual data range, and do statistical tests to see if two samples are significantly different from each other. If you look at in this particular figure, look at this the black error bars, right? You are looking at two different samples, right? And you are measuring a particular mean value for them, right? And what you do is you calculate their each samples average, right? This one here, this one over here, and then you calculate their standard deviations, and then you plot this 95% confident intervals by adding and subtracting two standard deviations, right? And that's how you get these error bars. And I will show you later how mathematically do that. Now, do these error bars from these two samples overlap? They don't. They come closer, right? But they don't overlap. When the confident intervals you calculate for two different samples do not overlap, right? That indicates that those two samples are significantly different from each other. It tells you that the mean value you were measuring was significantly different. Now look at this gray colored data set, right? So in this case, the confident intervals actually overlap between the two samples. Okay, so in that case, those two samples are not significantly different from each other, right? So now think again about means, right? If you just went by the mean, this mean and that they are not the same, right? So you would conclude, oh, because of the mean is different, those two samples are significantly different. Well, look at the ranges. Well, luckily for you, the ranges are also different, right? So in that case, it would have worked. Look at this case. The mean is not exactly the same between these two groups. They are quite different, right? This mean is slightly lower than that mean, but their ranges overlap. So despite the differences in the mean, because the actual confident intervals overlap, because the data spread overlaps, those two samples are not significantly different from each other. All right. So I did introduce you a lot of key concepts, but I'm going to introduce you a few more important concepts that you need to understand, at least from biological, at least in the viewpoint of biological sciences. Biological populations are highly variable. The only way to be absolutely certain of our conclusions regarding those populations is to census the entire population, to census all the individuals in that particular population, which is what? Impossible, which is not, logistically probable. So because of that, we use sample statistics to infer characteristics of that particular population, right? And we can use statistics to conduct hypothesis tests, and based on something called the rare event rule. This rare event rule is well explained in the lab manual, so I want you to actually read that chapter 2 and understand what that means. So if our hypothesis suggests that a particular event should not occur, but if you observe that event happening anyways, then the hypothesis is most likely false, untenable. As a result, if our results are so different from what would be expected, that it probably could not have happened by chance, right? Or probably could not have happened due to sampling error, right? Then we conclude that there is a significant difference, right? And because rare events sometimes happen, right? Rare events do happen, right? We can never ever be 100% certain of our conclusions. That's why we do not go more than 95% confident interval, right? We can never reach 100% confident intervals. Never, right? So because of that, because of there's an uncertainty, because things can happen by chance, because there is something called an inevitable sampling error, which will never be zero. In science, we never prove anything, right? We never prove that two things are statistically significantly different. We never prove that sample A is greater than sample B, right? So it's really important to know that. So never write we prove, they prove, or whatever, right? So prove it doesn't belong in the scientific world. So now organisms, animals or plants have actually habitat preferences, right? There are certain environmental conditions in which they do better, right? So there is, or in other words, there's an environmental tolerance, and you will learn more about this and niche, the environmental tolerance and niche in your lecture, right? So because there is a variable environmental tolerance, environmental preference among different species, rarely would you ever encounter a particular species being uniformly distributed across a particular habitat area. Because of that reason, when you sample, right, your samples should be representative of the population, right? In other words, you should avoid biases in your sampling design, in your sampling protocol, right? So this requires that you create, when you do a survey, that it requires that you create a sampling method that allows you to randomly select, right, a particular location in an entire habitat area, right? So in this case, normally what we do is, for a given area, let's say South Shore area, right, or Sandwich Beach, right, you kind of create a hypothetical grid over a map of the Sandwich Beach or Cape Cod Beach, right, and then you randomly pick certain grids, right, in that map to actually go and sample, right? Or you can actually, through a particular grassland area or a forest, you can actually create random transect lines and then walk along those transect lines while you are surveying, right? So there should be some sort of a mechanism where you create a way to randomize your sample, right? So that you're avoiding biases. In this particular exercise, in the activity, right now, this is actually by this level, you should have read the chapter, right? It describes how in a hypothetical bean community, you should be coming up with a sampling design to effectively randomize your samples to avoid biases. Okay, so now a bean community, is actually a box of beans that actually have 15 different types of beans. Each bean represents a particular species, right? And so you actually, if you were doing this in the lab, or when we actually collected this data, right, for you in the lab, what we did was we took a tray, a white big tray, right, and spread 15 types of different beans in that particular tray, right? And the tray itself is now broken into 12 cells or 12 grids, so by drawing 3 rows and 4 columns in the bottom of the tray, we actually created this 12 equal sized grid system, right? So now you have 12 grids, and your entire population is this all the beans that we have poured into the tray. Okay, now the entire tray has a lot of beans. You can never sample all the beans, right? You can never sample the entire population. So how do you pick samples? How do you, to avoid counting each and every bean here? Well, and I will ask you to select, let's say, 3 or 4 3 samples, right, out of 12 possible samples. So each cell can be a potential sample. You can derive 12 possible samples because there are 12 grids now, right? And we are not going to sample all 12, and you might just sample 3. So 3 is your sample size, right? Now the question is, how do you pick the 3 without a bias, right? You should not go for one corner and collect all 3. That's a bias, right? So you have to randomize the 3 grids that you are going to sample. For that particular randomization, what you do is you can actually roll a dice twice, right? First time, the die, when you roll the die, you select the column, let's say, or the next time you choose the row, right? Or you can, for the first, when you roll the die first, you can pick the row and then the column, right? It doesn't really matter. But in that way, you're actually randomizing, right? So now you are not picking a grid you like, you are actually picking a grid that you derived randomly by rolling a die. And rolling a die is an equal dice, equally sized cube. So so thereby you are actually randomizing how you pick what grid to sample, right? So so in this case, right, when we actually did the data collection for you, our goal was to subsample, right, at least 25% of the entire population of beans, right? And we are only looking at, although we have 15 different bean species, we are only looking at 2 different species: green lentil beans and pinto beans, right? Those are the 2 species that we are interested in, and we are only sampling 25%, right? So this is actually what the habitat actually looks like. The tray is our habitat, right? And all the beans are our community, right? So let me take my pointer here. Nope. Here. These are columns, right? And these are rows. So here you have 3 by 4, 12 grids, right? 12 possible samples you can collect. And from the 12 possible sample, you're only collecting just 25% of it. So imagine this 15 soap pack that we used is actually comprised of 15 different species, an animal community with 15 different species, right? A particular bean with a particular shape and a particular color representing a particular different species, right? And we need to have a way to sample this population. We have way too many animals here in this community, right? We are not interested in all the 15 different species. So what we see in this tray, that tray is going to be your habitat, right? The tray has that particular grid structure I just showed to you, the 12 grid structure, right? With the 3 by 4 gridded separation, which will help us set equal samples, unbiased equal samples will come out of these grid sets, right? So what we did was actually we make that tray like that. We created an artificial habitat, we created a sampling structure, and then we poured all that beans in this Tupperware into that particular tray like that, right? And then we actually, with that spoon, we made sure they are evenly distributed, right? So that you don't have clustering in this particular species in certain locations, right? And then we use this particular spoon to take samples, right? So what we did was a grid system, right? And with that spoon we collected one grid cell at a time. Before that, what we did is we rolled the dice, right? We picked a number for row and another number for the column, right? And then we determined what grid to survey and kind of collected all the beans in that particular grid. That's what we have done so far, right? So here what we have is actually, if I move over there, we roll the die, and then when you roll the die, if you get a number between 1 to 3, right, you have to get a number between 1 to 3 or to pick the row, and a number between 1 to 4 to get the get a column, right? So in this case we actually have we got number 3 and number 1, so which corresponds to the column 3rd and row 1. So what we did was we looked at the corresponding grid, and we extracted all the beans from that particular grid, right, back into this box, right? And then after that we actually started separating species in that particular sample, right? So in this particular sample what we are looking at is different species, right? So there are, like I said, the entire community had about 15 different species, right? And this is what we got in that particular first random sample. But we are not interested in all the species. We are looking at the pinto beans, right, and the lentils over here. These are the lentils and these are the pinto beans, right? So we started counting them, okay? And after the first sample is done, we roll the die again, right? And then we roll the die again twice to get what row we are going to look at and what column we are going to look at. Then by that one we pick that particular grid right over here and extracted that sample, separated the species and counted what we are interested in, right? And again, we did it for the third time. Sampled it, rolled the die, picked the random grid, separated this bean species, and counted the 2 species that we are interested in, right? And we did it several times, right? We did it actually 1, 2, 3, 4 different times, right? And created 4 different well, not 4 different times. We actually did it 3 times, right? We took sample 1, sample 2, and sample 3. That is roughly about 25%, right? And we created and we replicated that experiment 4 different times, right? So so that we actually have 4 different data sets. In each time we counted for each sample how many lentils were there, how many pinto beans were there, right? So now this data set, so is for you, right? We did the sampling, randomized sampling in this bean community and provided the data for you to continue with statistical analysis. So when you do this, right, pick any one of these 4 teams, right? Either you can randomly pick team A, B, C, or D, right? It doesn't matter what you pick. Don't do all of them, just pick one of the tables, one of the data sets, and continue on your experiment, right? So, and I will actually next I'm going to do a demonstration based on one of these data sets, right? I'm going to just randomly pick team D from here and use that data to demonstrate to you how to extract mean and other statistics, and how to ultimately, based on all the data, how do you run a statistical test. Okay, that's what I'm going to demonstrate to you with Excel. Right, again, for you, when you are doing these statistics, right, for the first part you are only using one set of the data. Okay,